differences to begin with, and that we can harness towards understanding, universal or unique aspects of cancer progression within our bodies. So, okay, maybe we can, for this one, we can dim the lights a little bit, because I will not use the board as if I can. We can avoid that. Yeah, awesome. Maybe a little bit more. Is the red visible? The red should be, no, not so much, right? Yeah. Can that be improved a little bit? Can you dim it a little bit further? Yeah, those two, yeah. Can these two be switched off and something else be turned on instead? Incremental improvements. Okay, I'll probably move along. So, as I was trying to explain yesterday, we were talking about a certain organ architecture that typifies how what we call homeostatic organs should behave like, a particular architecture where cells are expected not to move around, and hence the liver doesn't go and mix with the pancreas next to it. They retain a particular structure that determines the fact that they don't divide, that they don't move around. And then we also try to have an sort of overlay signaling / molecular map onto this that allows these molecules therefore work through the genome as well as the exterior milieu to keep this architecture in place. And that's what we typically refer to as an organ specificity, something that makes these organs, oh, now, okay, that's better, I guess. Something that keeps that organ, gives that organ a unique structure, and therefore its function. So structure in that sense determines function, and loss of structure therefore is concomitant with loss of function of that organ, which in another way can be called the cancerous transformation of that organ. And therefore, if one can now look at the arrival of this state of homeostasis as some kind of a endpoint of one trajectory, that trajectory is development. So you have therefore cells in a homogeneous state coming together through various mechanisms of pattern formation, of interactions with their extracellular cues, intracellular cues, gene expression, and the end point of all of this is this cellular arrangement, which we can call the homeostatic state of the organ. And in that state, the organ, now the cells are differentiated. In other words, they have reached a particular genomic biochemical state where they can be expected to perform that function that distinguishes them from other organs which have arrived at such a state using a different trajectory. Okay. So therefore, a developmental trajectory leads to an organ architecture, and the departure from this state therefore can be construed to be cancer, where now from this architecture, as this architecture sort of dissolves, as the rules of pattern formation are violated, now you start having depatterning. Those architectural arrangements no longer hold, and so you have a departure from this homeostatic state, and that departure would be your transformation. So homeostasis therefore stays in the center of where development ends and aging and other diseases and of course cancer begins. All right. So yes, Shradha.
Elaborate on that. And how do you say homeostasis is the fulcrum around which cancer was?
Sure. So as I elaborated yesterday, where development tends to end, mostly prenatally, that is inside the womb, or postnatally, which is after birth, is where you have cells coming together, interacting with each other, as well as interacting with their surrounding microenvironment, in, for example, the extracellular matrix, and reaching a state where they achieve a particular architecture, and that architecture allows them to now reach a state where they are no longer proliferative, where they are no longer migratory, and that architecture allows them therefore to function in that particular fashion. You remove any of these interactions and the state is no longer physiological, and that was what I was trying to elaborate yesterday in terms of cellular arrangements as well as the molecular underpinnings that lead to that cellular arrangement. So in that way, I would metaphorically call homeostasis as that emergent sort of outcome of developmental trajectories, and each of these developmental trajectories would be slightly different from different organs, which is why different organs look different and function differently from each other, and allow us to therefore specify, okay, these would be the gene regulated networks that have led to a pancreatic state, a beta cell within a pancreas behaving the way it does, secreting insulin, versus these would be the genes that led together to allow the cells to come together to form a breast, allowing it to therefore make milk proteins. Okay, well, this is awesome. I mean, I think this is the homeostatic state of the presentation slides. Okay, all right. Let's not depart from here. And therefore, that's the homeostatic state. And thereon, now you can introduce various aberrant cues either from the macro environment or chance events that now disturb the wiring of this homeostatic state, allowing you to depart from it, which is why you have these very, I would say, fanciful metaphorization of the similarities between development and cancer. People do, have this hand-waving aspect of trying to find analogies between that, because a homeostatic state being what it is therefore the end point of one and the beginning of the other. So, states, molecular or cellular, close to homeostasis on either direction would tend to look similar to each other. But if you were to go further down, you will start seeing differences, and that's when the thing. So therefore, one has to understand this particular process of what makes development or aspects of development somewhat close to cancer, and it's because of this particular way one can sort of arrange these states. And so now cancer therefore can be understood to be the loss of that homeostatic architecture, the loss of the rules perhaps that hold that organ together, the loss of those interactions between the microenvironment and the cell that I described yesterday, that entire molecular network, the loss or the breakdown of some of these rules would lead to cancer. And that's been kind of the mainstay of how one can define cancer in a cellular molecular context rather than just a molecular context where you talk about oncogenes and oncosuppressors, which are genes that get mutated, or a purely cellular way of describing cancer, which is like the unbridled loss of proliferation. So basically, one can therefore define now in this more complex way cancer as the loss of or the breakdown in the rules of pattern formation that led to that particular organ to come to be. And those rules would therefore differ when one talks about a prostate cancer versus a glioblastoma, a brain cancer versus a pancreatic cancer, and so on. But then, if one were to look again, one would take a naturalist stance and look at cells, and just look at cells and see how these cells behave in a cancerous state. One starts seeing that, and one sort of does not bias oneself against a developmental way of looking. Cancer and the way cells behave in cancer also seem to be adopting a new set of rules of pattern formation. And so one can therefore now think of this as cancer being an exploration of new rules of interactions. After all, cells also interact with each other when they are cancers. Cells also interact with their extracellular matrix when they are cancerous. And so therefore, one can sort of shift the definition a little bit and say cancer therefore is a re-exploration of new rules of pattern formation that may differ from the rules which were developmental to that particular organ. And if so, if we hold on to that thought for a bit, the question would therefore be, what are these new rules? And if we can understand these new rules, we can understand how cancer comes about and behaves the way it does. Okay. So this image sort of explores that thought, or gives us a visualization of that thought using breast cells. So we've taken breast cells, non-cancerous breast cells out here, and we've put them in this environment. The dark black basically represents the extracellular matrix that I was harping on yesterday, and within these, what we've done is we've taken a confocal image of that entire structure and then sort of flattened it out. So what you're seeing here is basically a rendering, a 2D rendering of a 3-dimensional structure. And there you can actually see how the cells are stained in red. Red represents the actin I was talking about, the cytoskeleton of the cell. You can see the cells are arranged in a particular concentric fashion and with a hollow in the middle, and then they are surrounded by the matrix outside. This is what is called an acinus of a breast. So in the breast, you would see that the cells are all arranged in this particular fashion, and that's what makes the breast a gland. A gland secretes, and so do these breast cells. So if I were to add a particular hormonal cue, in breast it is prolactin, a hormone that is secreted by the central nervous system, and that travels to the breast. That prolactin hormone, if I were to give this even in this setup in my lab, these cells would start making milk proteins. So the structure is also facilitating the function that it's supposed to be performing in the body. These are of course cells that I've taken out. I've taken the cells out, I've taken the matrix out, and I've reconstructed that entire structure in the laboratory under a microscope. Okay. So this is therefore the homeostatic organ architecture of the breast cells. If I were to, within the same environment, put in breast cancer cells, I would now see a departure from this pattern. The first thing is, these are cells which are cancerous but are not very aggressive cells. Yes.
So you put a collection of cells in this matrix and
Yes.
self-assemble.
They self-assemble, they self-organize themselves along with the matrix to give rise to these architectures. Absolutely. In this case, the cells do not organize themselves to this kind of a structure. What they form is a ball of cells which tends to divide very slowly over time. So you see that there is a loss of pattern, a loss in this particular arrangement, a loss in these rules of self-assembly, but it nevertheless forms a structure which one can define in certain quantitative metrics of visualization of morphology. And then if you take these aggressive cancer cells which are extremely proliferative and migratory in nature, you would see that these cells now start branching, showing branching patterns, and they branch out, show these long multicellular strands which are moving through the matrix, but in a way that I can perhaps again quantify the structure, put some metrics to it. It's not as chaotic as we think cancer to be. And therefore now can we also understand cancer as a pattern formation problem? Can we now lay down certain rules by which they interact with each other? Can we think that cancer too organizes, self-assembles in some way? Okay, towards that, maybe the arrow ones don't work, which frequently happens. Towards that, our group has been looking at this problem and looking at these rules, new rules of pattern formation. Incidentally, the new rules I would say is not a very new conceptualization. I don't know of how our forebears may have thought about it, forebears in an academic setting. Cancer is, in old textbooks you would often think of cancer being, you would see cancer being referred to as neoplasm. It's a term that is not used too much these days, but it is nevertheless referred to as neoplasm. And there must be some meaning to it, because neoplasm basically means new form. And new form comes through new pattern formation. So I think alternate interpretations of the larger understanding of cancer as being new form has been around since a very, very long time. Okay, so our group tries to look at these rules of pattern formation in cancer across different locales. And why is that there are different locales? Because unlike in development, where all the wonderful acts of the cell behavior and self-assembly take place in the same in that same locale, cancer seems to love to travel around. So cancer cells occur, or cancer cells tend to come together at what is called the primary focus, the place where you have the first cell that is transformed, and then therefore it starts growing out of that particular place, and that's called the primary focus. So one of the problems is early migration. How do cancer cells migrate out of their primary focus? The second thing what happens is they often tend to now find new areas to move to, and in order to do so, they try to get into the circulatory compartments of our body. These circulatory compartments tend to be blood vessels, lymph vessels for most, but also certain cavities in our body, one of which is called the peritoneal cavity, which is this abdominal cavity which is out here, which is again a negative space of all the organs that are present here. So if I were to look at image all the organs, all my abdominal organs, and give them a certain volume, I would still see that the abdomen consists of a large volume which is empty. That volume is called the peritoneal cavity. Similarly, we have something called the pleural cavity, which is out here, which is the entire thorax. Sorry, I have to use some anatomical terms, so I apologize for that. This part, the part of my trunk without the abdomen, this part above my diaphragm, without the lungs and the heart, there is actually quite a big space. Okay, these are very important spaces. They are very active spaces. They are spaces in which can get diseased very easily, and so on. And I'll show you instances of that. So anyway, so these spaces are filled with fluid. The abdominal cavity, that peritoneal cavity is filled with fluid. Pleural cavity is filled with fluid. So cancer tends to get into fluid spaces. So this is the process what is called intravasation, entry of cancer cells into fluidic circulatory compartments, survival within these compartments. And how do they survive within fluid environments when they are typically meant to be in solid environments, surrounded by cells, surrounded by matrix? Now suddenly they have found themselves in a new environment. How do they tide over such environments? What are the stresses involved? And finally, how do they tend to colonize new organs, to use a military term? How do they tend to decide which place to go to, settle down, grow and kill? All right. So we try to look at, as a group, we have tried to look over the last 8 to 9 years at all of these 4 locales and try to identify distinct rules, because each of these locales will have very different rules in which cancer cells are behaving with normal cells there or with extracellular matrix. I think there was a question, right? Yeah. Sorry.
Do these cancer cells, do they require a critical mass to start migrating?
Good question. This has been a matter of great debate for quite some time, because the simple sort of supposition for this was that yes, they do need it, and which is why there is a sort of a linearization in terms of the mass. You have maximum of it out here, followed by here, here, and then finally here. I think Mohit also referred to the process. Incidentally, this process is called metastasis. And Mohit was also talking about the inefficiency of metastasis, how a very few cells that actually get out from their primary locus end up out here. But there has also been some evidence against this linear idea, because it's known that even before this has developed considerably, some single cells can actually come out and populate other areas and actually grow even further than this process. So it's not quite clear regarding the critical mass. Okay. All right. I think there was one more question from someone else. So another raised hand. Okay, it's fine. We can keep moving. Yeah, please feel free to interrupt me as I go through, because there's a lot of biology here, which I shouldn't be apologetic about, but nevertheless, and so I can clear it as we go along. And so we've done a lot of. This is irritating. Okay, that's fine. And so we've done a lot of work. Oh, there is no. Yeah. The dongle was not attached. What happened to that nice circular thing that was there yesterday? Yeah, till then I can carry on. I still have about an hour and 10 minutes. Yes, yes, yes. Yes. Yes, to begin with, because it has to break through that cavity back into another organ, right? And I'll show you images and instances of that. But yes, of course, these cavities are also covered with cells for much part of it. Like the blood vessel, for example, consists of an inner lining of a cell called endothelium or endothelia. And so when the cancer cells ultimately want to now get into, say, the liver or the bone, they have to break through this cellular barrier one more time and then get into that particular organ again. So yes, there's a breaching of the cellular layer as they enter into the liquid zone, and a breaching of the cellular layer as they exit the fluid zone to reach another organ. Sorry? Yes, yes, yes. That is that is the predominant mechanism of entry into organ. There are other mechanisms by which cancer cells can also sort of creep and move as a mass to the, say, the neighboring organ without needing to jump into a blood vessel. So that can also happen. For example, in ovarian cancer, there's some literature that the cancer cells from one ovary sort of keep moving, walk over the uterus and go to the other ovary as well. Not very common, but it can happen. We have the blue. Still here. Beautiful, thank you. And yes. Okay, great. The question again, what are these novel rules of cell ECM interactions that govern the kinetics or even the happenstance of metastasis? How do these rules allow the cancer cells to metastasize or not, and also how fast or how slow? Because the kinetics of metastasis is very important as well. We often see that in some patients afflicted with the same cancer, they tend to die faster. In other patients tend to live long enough. So it's very important, therefore, to even understand what determines the speed of or efficiency of some of these processes. So we've done a lot of work in this a particular zone, trying to understand how migration takes place, what kind of rules of pattern formation prevail in this area. Mohit also briefly referred to some theoretical explorations in this process as well. But I will not talk about this published stuff, so we can, I can refer to you to the papers if you are interested. We have very recently entered into this zone as well, in building certain organ-on-chip devices where we can visualize the entry of cancer cells into fluidic compartments that we have built and rigged with microfluidic devices in order to see how flow helps in this entire process of intravasation, because this is very difficult otherwise to visualize in an animal, and so we've just gotten a patent on this device as well. But I will for today's talk discuss findings, some early papers as well as unpublished work on the process of migration as well as colonization, and certain interesting problems in these two zones. So how do cancer cells move within such fluid environments? I think it's a good segue from the question that you asked. And how do spheroids survive such confined travel? So for that, we'll just start by water. This is a problem in ovarian cancer. Okay. In fact, I think all the 2 or 3 problems that I will discuss today refers to ovarian cancer. So Okay, here is the abdominal cavity. As you can see, this pink zone is that sort of negative volume, if that makes sense, of which is basically the space which is left behind after all the volumes of the different organs in the abdomen are accounted for. This is the liver, the biggest organ in the abdomen. Here you have the stomach, and the intestines. The intestines are all basically moving in a left-right in sort of inside-out manner. So you can see the cross sections actually out here. So as you can see, they all are floating within this cavity, and so this cavity is called the peritoneal cavity, and that is the battleground for the ovarian cancer. So what happens is, out here, this one is the uterus, and next to it are the ovaries. And so cancer cells from the ovaries jump into this cavity, float around, and eventually latch on to these organs and surround these organs. So what you have here is metastasis that is slightly local, well, not local, but I mean it happens mostly intra-abdominal. It moves mostly in the abdomen. It doesn't go to the brain or to the bones, unlike some other cancers do, because I mean it's able to colonize the abdominal organs and do so efficiently. Now, when these cancer cells move into this cavity, fluid-filled cavity, it's a fluid-filled cavity. When they move into this fluid-filled cavity, they tend to form clusters. So, typically you would again think of cancer as a unicellular phenomena, as I explained yesterday, single cells, EMT and so on. Here again you have a collective behavior coming through. So, cancer cells within fluid environments tend to form multicellular clusters. These clusters are often called spheroids because of the shape that they assume, almost a spheroidal shape that they take. Now, these are images, bright field images of the cells as they were taken from a patient. The first patient that we could get some fluid from this fluid of an ovarian cancer patient in a hospital in Bangalore. So when we are looking at these cells, the cells that had sort of metastasized into this peritoneal cavity, that were floating in this cavity, we did see clusters, but something struck us as very interesting, and that is that these cells had formed a cavity. So they had, in other words, self-assembled themselves into a cavernous or a cavitational or a luminous architecture. So as I explained yesterday and early today, you typically have a departure from a homeostatic state. For example, in the breast, where you have a hollow in the middle where milk is supposed to be secreted, cells arrange themselves in a hard cell-cell adhesion dependent manner and so on. Here you have cancer cells which have escaped out and are forming and are self-assembled to form these kind of structures. Okay. So a vindication of the idea that even cancer cells can self-assemble and form patterns and form organizations. Okay. Great. So we did find both solid clusters, cells that were clustered together to form into these sort of grape-like structures, as well as hollow structures. And in different patients, we would see different proportions of these two different types of spheroids: solid spheroids, hollow spheroids. Hollow spheroids or hollow arrangements of cells to any developmental biologist worth a salt is a semblance of polarity and organization. Okay. So this got, of course, this got us excited. Yes, Ronnie. Okay, I can come to it towards the end, but hollow ones tend to, patients with a greater proportion of hollow ones tend to do worse.
So is it that like the cells in the hollow spheroid thing, they have a lot, like sort of differentiated on the surface?
I'll come to that. I'll come to that. I'll come to that. These are exactly the kind of questions that intrigued us. The question is to a cell biologist would be how. How do they form these structures? But to a say more of a morphologist would be, or a developmental biologist would be why. Why the heck are they even forming these two kind of structures? So interestingly, again from a cancer biology point of view, you have this very interesting topic in cancer which is called tumor heterogeneity. The fact that you have different cells behaving in different, showing different behaviors coexisting together. And I think Mohit may have referred to that in terms of heterogeneity and plasticity. I missed the first lecture, so he generally talks about that. But sorry, yes. Sorry?
The hollow spheroids always form from the solid ones.
Great question. I'll show you that. Yes, they do. They form from the solid ones. Yes. Yes.
We talk about the metastasis process.
Yes.
Then it will be good if the cells are in the cellular form other than tissue form, because it has to again reassemble and then invaginate to other space.
Right.
organs.
Right.
Then why they are forming?
Why? That's the question. Okay. I'll tell you why they are forming that.
Okay.
Okay, in just a bit. So we saw that. So this is also intra-patient interspheroidal heterogeneity. So in other words, you can not just have cell heterogeneity at the level of cells, E or M, proliferating versus migration, and so on. Some of these binaries that Mohit talked about, but you can also have heterogeneity at the multicellular state for cancer. It's a very interesting concept. Okay. So cancer cells within the same patient can form two different types of multicellular organizations. Again, question why and question how. So we try to replicate these things within the laboratory. We take cells, we get them into a setup where they are now floating around in fluid, and when they do that, they tend to form these kind of solid structures. We call them now moreuloids. Because biologists always love to invent new terms and complex terms to describe what they want to do, but also because of again this parallel with embryology or developmental biology, which I'm never able to escape personally, and because this particular shape is Latin for mulberry or bunch of grapes. And therefore embryos at such a stage are called moreulas. So we call them moreuloids or morula-like. And similarly, there is a state in the developing embryo called a blastula, where you have cells arranged in this fashion with a hollow in the middle, and so we call them blastuloids. So we saw that cells first, when cells are de-adhered from being adhered onto the substrates on that plastic that I was referring to yesterday, they form these kind of structures, and then over time they give rise to these hollow structures as well. And so the question is, of course, why and how? So this is a scanning electron microscopic image of the solid cluster. So as you can see, it resembles, to anybody who's seen embryos in this state, it does resemble a morula uncannily. And then this is the hollow spheroid, the blastuloid. Now it's difficult to see the lumen, of course, because we are imaging the entire structure and looking at the outer surface of the structure. But one interesting feature that distinguishes this from this is the fact that in case of morula, you can see the furrows between the cells very clearly, whereas here it seems as if there is a coat of something that's covered the cells, and so you cannot see the distinguishing furrows between the cleaved cells, between the dividing cells. And so what was this coat? Again, looking at the structure and understanding this in more details, doing the usual stuff that we do, staining the cells staining these structures for proteins and all, we identified that this coat is actually what is called basement membrane, or the same extracellular matrix proteins that I was talking about yesterday in an organ homeostatic context. In other words, if you remember that coat that is present on the top of cells, the ECM integrin that decides to turn down MAP kinase, that decides to decrease proliferation and migration, that coat was now present on cancer cells as they form these multicellular structures. Recognize the opposition of these two statements. Cancer cells, yesterday I talked about how you get in cancer, you get the breakdown of the matrix, and therefore turning on of the signaling that leads to proliferation and migration, right? Here, instead of breakdown, you have synthesis of that extracellular matrix. So cancers can also, if required and under certain contexts, give rise to a homeostatic architecture in order to perhaps overcome some requirements or some stresses in certain microenvironmental contexts. Because remember, cancers travel, so they have to tide over different stresses in different microenvironments. What are those stresses? I'll come to that in just a minute. So just again, cancer cells, these are spheroids. Spheroids which are, especially the hollow spheroids, have managed to form a matrix around them. So when we talk about these multicellular clusters, they are not just cellular clusters, they are almost organ-like or tissue-like structures which have the cells and have the extracellular matrix. The same kind of binary organization that tissues and organs have in our body. Question?
So both morulloids and blastoids, both of them secreted EC?
Okay, I'll come to that. I'll come to that. So do both morulloids and blastoids secrete the extracellular matrix? No, to a large extent, well, yes, to a large extent, yes, but their organizations were very different. In case of morulloids, this particular protein called laminin, which is one of the major proteins of that superstructure, was present more in a cellular fashion, when in case of the blastoids, it was present on the surface, just coating that entire surface almost like an organ, almost like that acinus that we talked about some time back. And this was true for patient spheroids as well. Now, so the question that you asked, is the extracellular matrix secreted by both morulloids and blastoids? So for that we did again something called quantitative proteomics, where we looked at what are the different protein signatures that are typical to blastoids versus morulloids, which proteins get enriched further in the blastoids and are not present in the morulloids, and vice versa. And that led us to then this understanding that no, indeed what you are saying is true. Perhaps both these spheroids do make extracellular matrix proteins, but of a different types. So again, harking back to the idea that different organs will have different rules of pattern formation, will have different components giving rise to the pattern formation, will have different differentiated cells and perhaps different extracellular matrices that make those two organs different. So it is true for these two cancerous multicellular clusters. Morulloids make different extracellular matrix proteins than blastoids, and hence their form, their pattern formation rules must be different from that of blastoids. And indeed, morulloids make more of a protein called fibronectin. Blastoids make more of extracellular matrix proteins called laminin. And the two have very distinct functions in terms of how they modulate cell behavior on them. Classical cell biology literature shows that on fibronectin, cells tend to move. On laminin, cells tend to stay still. That's why in organs, in homeostatic organs, you don't get laminin. So you don't get fibronectin, you get laminin. On the other hand, in developing tissues, you have more of fibronectin, because in development, before you reach the homeostatic state, in development, cells move. You have extensive cell migration, and that's how we get our body. So again, you have developmental rules coming in and getting recapitulated in cancer. So there's this famous quote that ontogeny recapitulates phylogeny. Here I can turn it around and say oncogeny recapitulates ontogeny. Cancer recapitulates aspects of development. Aspects of development.
Sorry.
Yes.
So in both cases, the individual cells are identical, but it's just that the shape determines the molecules that are secreted around.
Yes, you nailed it. In both cases, the cells are very similar. Well, similar as in they are the same. But of course, they are, the matrices are being made by the cells, so that their biochemical states are now slightly altered. Okay, so after a while, the same cells which were part of the morulloid structures have now formed blastoid structures, and that is again an outcome of their interactions with the matrix that they form in the morulloid state. So just the way you have development going on, here too you have a developmental, two stages of development going on even in case of the cancerous cells. A developmental stage 1, which is a morulloid developmental stage, to a blastoid developmental stage. All right. Okay. why. So we are now inching towards the why question here. So, but before that, like, so what makes a hint at why? What makes blastoids and moroloids different in terms of their behavior? Now, they look different, fine. They have a hollow, they don't have a hollow. What makes them different in terms of how they behave? So to understand that, what we did was we took these moroloids and bombarded them with single cells to see how transient versus how stable these structures are. And we found that moroloids tend to accept cells when present around them in this fluidic state. Moroloids also tend to coalesce and form, they almost behave like liquids. So if you have two liquid droplets coming together, you would have a bigger drop, and so do moroloids behave similarly. When you have two moroloids coming together, if you jam enough number of moroloids within a tiny fluid compact volume, the moroloids would tend to come together to form a single moroloid. Okay. On the other hand, blastoids, the ones that are the rounded hollow structures, tend to be extremely stable entities, almost organ-like. You put cells around them, they don't accept them. And I bet you cells won't even be coming out. It's very difficult to see cells coming out because we do this by looking at cells or these structures every few minutes. Now a coming out event is very difficult to sort of visualize, but anyway, but going in is something that's easy because we can fluorescently label cells and then see whether they have been taken up or not. So we do see that the hollow structures don't tend to accept any single cells around them. They don't tend to coalesce to form bigger blastoids. So blastoids are stable entities. Moroloids are volatile, transient entities that tend to be morphogenetically more labile rather than stable. They tend to be loose in terms of whom they join together, whom they accept, whom they reject, and so on. Blastoids are much more organ-like, homeostatic-like structures that tend to exist. Okay, are these stable structures? Are the hollow structures, the blastoids, are they like balloons that are just floating around in the fluid? No, they are dynamic in terms of their structure as well. How do we know that? Okay, no, this is the wrong time lapse video. So here is where, sorry, this result is where we asked for blastoids, is that basement membrane matrix, that extracellular matrix that we described, the laminin, is it required for the structure or not? So we used an enzyme, and when we to basically remove that coat, that coat that surrounded the blastoids, and when we used that enzyme to remove the coat, we saw that they moved very quickly into once again a grape-like entity. And if I can play this once again, I think it should play again. When we washed out that enzyme, those moroloids again recapitulated into blastoid shape. You can see this blastoid coming back again. So in other words, that matrix was not just present and was not just characteristic of the hollow structures, it was also causal to the hollow structure. That matrix is essential for that morphogenetic state, that state of being a blastoid as opposed to a moroloid. And if you were to remove that, using an enzyme, if you were to remove that coat, it moves back again into a moroloid state. Yes?
In the beginning, you mentioned that there were different proportions of moroloids and blastoids in different patients.
Yes.
So can we say the patients who had a much higher degree of cancer, they had much more matured blastoids than moroloids?
That is one, and I'll also give you another explanation of why the patients may have had a more predominant proportion of the blastoids. I'll come, just hang on for just a couple more slides. Yes?
Just a small clarification. These moroloids and blastoids, if we put a cell, basically cells in a in vitro moroloid, will that transform into a blastoid later on?
What do you mean by in vitro? These are mostly what biologists would call it in vitro experiments. These are not happening inside a patient.
So I thought you are taking at the different stages from the patients.
No, we can't take it at different stages from a patient because these things, this removal, the patients can be subjected to such removals on a regular basis. The doctors take out these from the patients only once when they come to the hospital with sort of a fluid filled abdomen. So the fluid is taken out at that stage, and so we can at best access it at that stage. So that's it. But much of the understanding of how they sort of move between each other and all are mostly at the level of in vitro, because that's what we can do, or from mice, which we have also done this within mice, which can sadly be subjected to more of these kind of interventions than humans.
Okay, thank you.
Okay. Going back to that architecture question, I'll sort of try to breeze through this now because of time. And also because I described a little bit of this yesterday. Now remember the whole idea of the organ architecture and how it forms through an interplay between cell ECM interactions, which lead to the signaling state getting into a homeostatic state, which not just involves our favorite MAP kinases and PI3 kinases and so on, but also includes the cell-cell junctions, allowing the organ architecture to therefore have not just cellular properties but tissue level properties. We looked at those also in this context, asking, okay, simplifying this point, asking whether the cell ECM interactions or the presence of the ECM out here also drives the intercellular arrangements within blastoids or moroloids. Okay, that's the question. And we looked at this particular molecule called ARP23. Again, the name is not important, but ARP23 is involved in this formation of these cell-cell junctions, and we saw that ARP23 was present to a very strong extent between the cells in case of the blastoids, and if you removed the extracellular matrix, you would tend to lose the presence of the ARP23. In other words, yes, just like organs, just like normal developing organs, even in these spheroids, if you were to remove that basement membrane of the blastoids, you tend to lose the cell-cell junctions that are also, or the cell-cell junction proteins that are present between the cells in case of the blastoid architecture. And you can see how even the architecture has gone from having a lumen to losing that lumen. And if you were to use an ARP23 inhibitor, a molecular inhibitor that actually breaks down this intercellular junctions, you tend to actually form extremely bad moruloid-like spheroids. Here are the nice blastoid spheroids. You have now lost them and form moruloid spheroids. Okay. In fact, you get very poor spheroid formation if you were to add the inhibitor even before the cells have come together. So these are in vitro experiments that we can only do in a flask in the lab, right? And other control experiments. In fact, ARP23, the one that gives rise to these kind of structures, leads to loss of cadherin. Remember cadherin? That is essentially the intercellular glue. That intercellular glue is also lost when ARP23 has been inhibited, and it leads to loss of another protein called ezerin, and ezerin, which allows that cytoskeleton to link up to the membrane underlying the cells. So in other words, it gets the cytoskeleton to be at the right place. And finally, you also tend to lose this expression of the same matrix. So matrix makes ARP23, makes intracellular adhesions, and gives rise to the architecture. You lose the matrix, you lose ARP23, you negate ARP23, you lose the matrix. So you complete the loop, in other words. So the intercellular junctions and the cell matrix junctions reinforce each other. The same point that I was showing, that feedback that I showed that gives rise to this homeostatic state, is being recapitulated by cancer cells. Remember, in cancer you tend to, when you talk about the mesenchymal state, you tend to talk about the loss of cell-cell junctions, right? You tend to talk about the loss of E-cadherin, which is your standard epithelial marker. Here are cancer cells which are floating around the body, which have recapitulated the E-cadherin, which have recapitulated the glandular architecture altogether. Okay. So the question of why do we need blastoids and moroloids? Why is it that patients tend to have a preponderance of the blastoid state? To understand that, let me just finish with this one and then we'll go. To understand that, what we did was we tried to look at these structures in the context of how they are able to attach, because eventually all the spheroids have to attach onto biological surfaces which are present inside the cavity. And we saw that moruloids tend to attach much faster, and that gives us a clue, because in a patient who typically comes to the hospital only in advanced stages, Indian women coming to the hospital is a rarity unless they are pushed to come to the hospital. That's the a reflection of our socio-political state. And so unfortunately when they come, the cancer is already at an advanced stage, and at that stage most of the moruloids will have attached, leaving the cavity full of these blastoid structures. So blastoids tend to stay in the abdomen for a very long period of time. Moruloids get formed and colonize very fast. So they are early colonizers, poorly stable or labile entities which tend to colonize fast. Blastoids on the other hand are long-term survival strategies that cancer cells mount towards surviving within the patient for a very long time. To the extent that, and here is where we again resorted to microfluidic experiments, where we tried to mimic how cancer cells, how such entities can move through tiny spaces that are present within the abdominal cavities where they tend to move to and finally colonize. We saw that moruloids, moruloids, as they move through such volume confining environments, tend to deform and form more of these sorts of sausage-like structures and tend to break down. They tend to disintegrate very fast. Hollow spheroids, the blastoid spheroids on the other hand, because of their structure, behave like what are called poroelastic materials. They tend to drive out the fluid as they move through such confining environments, and once they come out, they inflate back again. Okay. So they are poroelastic type of materials that are able to tide through such volume confining environments which are present in the abdomen, and are able to move into tinier spaces where they can colonize. So they are less adhesive, stable, resilient multicellular structures that can exist within patients' cavities for a longer time, which is unsurprisingly why when we take out the fluid from patients, we see the fluid chock full of these kind of structures rather than these structures which have already attached. Okay. Indeed, these structures have, because they survive long within the cavity, they have many other interesting attributes, ominous attributes to them, which allow them to survive chemotherapeutic insults and so on, but that's a different story. I'll tell it at a different time. So in other words, you have, I missed a summary slide here. In other words, two different spheroidal forms can exist within the same patient with distinct behavioral profiles, allowing cancers to metastasize fast or allowing cancers to metastasize on longer time scales. And which is why ovarian cancer, typically when detected, one can use chemotherapy and one can sort of, in surgery of course, and one can remove the cancer mass easily, but it invariably tends to come back again and again, because there are these cellular populations that tend to survive long periods and tend to hide within tinier compartments within the body over long periods of time. Okay, all right. Second story. I have to 11:30, right? Yeah, awesome. Yes, please.
So when I teach ecology, I often talk about these two possible life cycle strategies that species have developed. One is you produce lots of offsprings, you don't invest too much in parental support, they let them sink or swim. But because you're producing in such large numbers, you hope that some will survive. The other strategy which humans have adopted is have a few, but invest enormously in enabling them to survive up to adulthood, and let them, so a few, but those few will prosper.
Yes.
So is this kind of the difference the difference that you're trying to illustrate between these two?
Yes.
Okay.
Yes. So this is exactly, I mean, the motivation for looking at this came from my readings of ecological strategies, and these are trade-offs that even the cancer seems to compute as a population. to devise these strategies towards two different kinetics of survival. It's amazing how they are able to do so. And yeah, absolutely, the parallels are very strong. In fact, there are again, I would say horribly interesting parallels between cancer and ecology on some other problems also where we look at, where you have, for example, resistant cells, drug resistant cells and drug sensitive cells, and how they interact with each other. Maybe I can talk about that later. Yes? Can these structures multiply on their own? No, sadly they are not. They are organ-like but not organism-like. They've still not reached that stage. So they can't multiply on their own, but there's not much cell division that is actually occurring inside such structures. And you would understand why, because if you have cell division typically happens when cells lose their contact with the surface. So if they start dividing, they will actually break down. So they don't tend to divide. So this is a period in time where they are surviving through this fluid environment. They typically again tend to stay quiescent. Okay. Yes. Yes.
Is it capable of converting back to the monocyte stage?
Very, very good question. Is it capable of converting? Capable? Certainly. And now we know at least 2 or 3 interesting biochemical cues that can convert them. One you just saw, that collagenase enzyme that I gave converted them back to moroloids. What is that collagenase enzyme? It's the same enzyme that I talked about yesterday out here, the MMPs, which are the protein cutters, which is responsible for converting a normal state into a cancerous state. That same enzyme is employed here to convert, can be used in this context to convert a blastoid to a moroloid. If you use that ECM breaking enzyme, you can immediately convert them into a moroloid state. In fact, I suspect, we've not proved this at any point of time, but I suspect what might be going on is when the blastoids don't tend to attach, right? But when they come eventually close to where they are supposed to attach, you would have higher levels of this enzyme building up, leading it to become moroloid and attach immediately. But we've not proved that.
So this is like I'm basically making an analogy between the blastoid stage and a spore. So
yes.
So is a blastoid basically when the initial proliferation of the cells will happen, it will basically encounter a resource-rich environment. It will multiply, and then it might move out.
Yes.
As the resource decreases, it
Yes.
might convert into a blastoid stage,
Yes,
and the
yes,
moroloid
yes.
will move out.
Yes.
Then, so now the removal of moroloid is happening because of treatments and other things.
Yes.
So then it is useful if blastoid then get converted into moroloid and
Yes.
then basically colonize other parts
Yes.
of the body.
Yes.
So is it able to basically, is there a signaling happening so that blastoid get a signal so that when it needs to convert to the moroloid?
Well, yeah, exactly. We have not yet at least shown any evidence of that signal, or we have not found any evidence of such signals. But there is literature, there is some work in the literature that's sort of in the field that shows that what you can have is, not in a direct fashion, but in an indirect fashion. When say if you have a spheroid or hollow spheroid out here, for example, coming close to a peritoneal cavity where here you have cells, what can happen is, while the spheroid will not make its own enzyme, what it can certainly do is it can induce the production of enzymes from fibroblasts and other immune cells, which in turn will start making this enzyme, and that enzyme will now convert the blastoid into a moroloid state, allowing it to attach faster. Okay, this is possible. Oh yeah, okay. This is possible. So the cancer can induce cells which are close to where it has to colonize, to in turn make the enzyme that can convert the blastoid into a moroloid, allowing it to attach. In the absence of this cue, or when it's far away from the surface, this enzyme will not be able to convert it into a morluloid state, allowing the blastoid to survive within the environment for long periods of time. So this is possible. This is just a thought model. We have not explored this. We have not found any evidence for it. But yeah, this has crossed my mind, of course.
Yeah, because if we can somehow prevent this signaling from basically giving the signal to the blastoid of conversion, it can basically, if blastoid is staying in the body and not converting into a moraloid, and if moraloid are absent, then it does not basically that harmful to the body, and it can be kept in a quiescent stage.
Agreed. But having said that, remember, I was just talking about the decreased attachment of blastoid relative to moraloids. Ultimately, they will attach as well. So it's not that they never attach. They don't necessarily need to become a moraloid to attach. They will attach. It just they take much longer periods of time to do so. Okay. It's a relative term. Yes.
So these moruloids and blastoids are made up of the same type of cells, right? So essentially they are genetically identical.
Yes, genetically
but,
identical.
As you showed, like for example in the case of fibronectin and collagen, laminin, gene expression levels are changing.
Yes, so epigenetically.
Epigenetically, exactly. So my question is like, so you showed the time lapse where a moruloid was becoming a blastoid, right? So how does a cell in a moruloid know that it should change its expression patterns?
So as gene expression takes place on very rapid time scales. So what I showed, the time lapse that I showed was on the order of, you can actually turn back the light. I'm hopefully done with the board. I'd never know. Anyway, so gene expression changes happen very fast.
Yeah.
The time lapse that I showed is on the order of hours, about 6 to 12 hours.
Right.
So it takes about 12 hours to shift, and you can get gene expression changes through signaling occurring within minutes. So it's very easy for, once you have that loss of collagen because of the collagenase, that again switches the signaling state also very quickly into a stage where now you have the loss of the basement membrane matrix. The loss of the basement membrane matrix in turn turns on fibronectin expression, and so now you have the laying down of fibronectin and moves into a moraloid state pretty fast.
I'm talking about moruloid to blastoid.
Oh, moraloid to blastoid. Moraloid to blastoid takes much longer time.
So what are the signaling cues which convert one moruloid into?
What I showed some time back. So what you have is the moraloid state exists and is predicated upon the presence of fibronectin. Fibronectin over time now goes down. Once fibronectin goes down, the ARP23 signaling goes up. ARP23 is driven by something called the Rho kinase. So once ARP23 and Rho kinase go up, you have the laying down of the laminins on the cell surface and the cadherins between the cells, and that sort of puts in the cell-cell junctions and the cell matrix junctions in place, allowing you to sort of move from the moraloids to the blastoids. Of course, I have not yet explained what leads to that cavity. The cavity is driven by channels, sodium channels, that now allow the fluid to be pumped in, leading to these kind of structures.
Just one more.
Yes, of course.
So this fibronectin levels going down,
Yes.
you mentioned.
Yes.
So this happens in all of the cells, right? So how
Yes.
does it? So does it happen in a coordinated fashion so that it converts into a
So,
blastoid?
yeah, that's a great question. We've not looked at it. We've not looked at whether there's synchronous decrease in fibronectin, because we tried to track it at the protein level, and fibronectin being an extracellular matrix protein tends to come out of cells. So
Right,
once it's
right.
out of cells, it's not easy for us to even know whether all cells are not making it now or whether some cells are not making it and so on. But we know that the level of fibronectin almost acts as a gatekeeper for whether it remains in the moraloid state or blastoid state. In fact, if we decrease fibronectin, it switches very fast into a blastoid state.
Thank you.
Thanks for the question.
These blastoids are more of a localized state than compared to moraloid.
What do you mean by local?
Localized in the sense they don't move that much compared to.
No, they're floating. They're floaters. These are passively floating around in the fluid, or passive is what I think they are. Passively floating around in the fluid. So there is of course a convective current of the fluid, because this fluid is filtered from blood. So you have fluid getting filtered in and fluid getting filtered out. So that creates a convective current, and that allows these floating cells to therefore sort of keep moving in the abdomen like this.
But they are localized to the abdomen.
They're localized to the abdomen.
So in that sense, like targeting blastoids should be easier compared to moroloids, because the moroloids can basically travel through the blood vessels and move into other
No,
parts of the.
on the contrary, we think that the blastoids are moving to the blood vessels, because the moroloids, as I said, tend to attach very fast. So the moment I shed it into the abdomen, they settle down somewhere in the abdomen and start dividing. The blastoloids are able to move through those confined channels. Those confined channels are the way through which this fluid is getting filtered into blood. So they basically are getting stuck in that gutter through which that fluid is getting out, but they are also able to deform and move through that gutter and come out to the other end. So that allows them to actually get into blood as well. So that's what makes them more dangerous.
I think for like the transition between moroloid to blastoid, the environmental cue will be the channel, like the transfer, the metastasis of that cell from one organ to other organ. So they have to squeeze into it.
Yes. But they have the ability from their material properties. Their rheological properties allow them to squeeze. What allows them to get attracted towards those specific organs? Okay, maybe I'll talk about that in the second part. I think I won't be able to get time for the third one. That's okay. So here's the question that you kind of asked and framed in a different way. What determines the colonization of these cancer cells in new stromal locales? Okay, so this story is where we sort of started looking at the cancer cells as they start invading this first layer of normal cells that they encounter at the border of the abdomen. So this is the abdominal frontier, where now you have this spheroid coming in, and how does this spheroid sort of penetrate through this barrier to start now colonization? So we quickly found out that there's something very interesting going on when cancer cells encounter such normal cell monolayers. Then it's not very easy to penetrate normal monolayers, even if one can think of cancer cells as these odious ogres which can penetrate and kill normal cells. The normals, while the spheroid, this is a spheroid that we put up and looked at how it sort of behaves in the context of the normal cells under it. I hope it starts again. Yeah, there you go. So this is a spheroid, and while the spheroid settled down and started attaching, very soon we found that this spreading sort of stalled, and the normal cells around it are actually able to push back to the extent that they would keep the cancer cells localized where they first settle down. So it's not very easy for cancer cells to even break through a single monolayer of cells. Forget the immune cell, cancer cell aspect, and immune defense, immune evasion, and so on. Even simple normal epithelial monolayers can very well defend themselves against an onslaught of cancer cells. And so that led us to ask, therefore, what might be the cues in which such defense can now be quelled by cancer cells, or under what situations is this competition between cancer cells and normal cells sometimes tilt in the favor of cancer cells or in favor of normal cells. To understand that, we got into the question of aging, and age, as we know, demographically or epidemiologically, people who cancer tends to occur more and tends to progress to worse at a worse kinetics or at a higher kinetics in aged populations. Aged populations tend to get cancer more, tend to do poorly when cancer afflicts them. And so a cell biological understanding of this it typically involves immune system must be therefore not very efficiently performing in older individuals than in younger individuals, and so on. But we ask the question of whether this particular aspect, whether the defense that normal cells can mount against cancers is also weakened in older cell populations. And towards this, what we did is that we started making these sort of co-culture competition setups, where we had cancer cells, in this case called SCOV3, on normal cells which are in this case called MET5s. These are mesothelial cells, or cells that are typically coat the abdominal cavity. They are the first layer of the abdominal cavity that cancer cells encounter. And the cancer cells were stained in green, the fluoresced green, the normal cells fluoresced red. And we saw that when the normal cells were younger as opposed to when they were older, in which case what we had done was, in this case these normal cells were rendered what is called senescent. So senescent cells are cells which now are cells that have either been exposed to radiation or to multiple rounds of cell division or to certain debilitating chemical agents like chemotherapeutics and so on. So they tend to resemble cells which in older individuals. And so when cancer cells were with normal epithelia as opposed to aged cells, the cancer cells tend to grow to greater extent. To understand this process of why that was happening, we then started looking at the interactions very closely, and we found that cancer cells to begin with tended to surround those normal cells. The cells, again normal cells are in red here, tended to surround normal cells which specifically were more senescent, because whenever we tend to induce senescence in a population of say 100 cells, not all 100 cells would become senescent. If that were the case, we as aged individuals would have completely dissolved. So we always, there are cells within our tissues which are slightly more older than cells which are younger. So within such mixed normal cell populations where there were these aged cells, cancer cells tended to come close and stick close to these aged cells, and they were found to be more localized towards the aged cells. Now, was this enough for the cancer cells to start growing to a greater extent? Was this localization happening in an undirected fashion in which cancer cells would come and would accidentally be found near the aged cells and then would move on? To understand some of these processes, what we did is we used an agent based model in a framework called a problem solving environment called CompuCell 3D, which is based on the cellular POTS model, to devise a series of interactions between cancer cells, young cells and aged cells. So in this 3-cell population, we tried to look at what are the interactions which would lead eventually to the effect that aged cells tend to get removed from cancer cells. In this process, we will now go in a bottom-up fashion to now design an iterative set of rules by which this pattern formation can be studied. So instead of breaking things down from an experimental side, using simulations, we can now construct rules from bottom up to see how the end sort of comes about. What are the emergent outcomes of these rules in such simple computer settings? We had about 4 models, and the 4 models were as follows. The rules that were specific to these cells were that in case of model 1, which was the simplest, the vanilla model, we basically had cancer cells that would not be localized around the senescent cells in any way. So that there's no interactions between cancer cells and the senescent cells. In model 2 and 4, we had 2 interesting rules that were going on. In one rule, we had the cancer cells localized closer to the senescent cells, and if they are localized, if enough number of cells are localized close to the senescent cell, the senescent cell would get extruded, allowing for the cancer cells to now grow. This is precisely what we saw in our previous experiment. The second rule that we imposed was something interesting, and that is that cancer cells also tend to have a higher degree of adhesion with respect to the senescent cells. So there was a greater adhesion, not just localization, but greater adhesion to the senescent cells. And we saw that this rule was quintessential for us to be able to get to a state where senescent cells would eventually get removed. The big red cells are the senescent cells. The senescent cells would get removed, and the cancer cell would reach a population where the cancer cells would eventually have a higher degree with respect to the normal cells that were present. So the ratios was closest to what we got in experiments. therefore, that predicted that not just were cancer cells localizing close to the senescent cells, they were also attaching, they were adhering more better with the senescent cells. So, there was a strong adhesion force that is also driving this particular process that was involved in the cancer cells coming to the senescent cells and removing them. What was this adhesion force? To understand this, what we did through a series of experiments was we took cancer cells, we took these mesothelial cells, aged cells and normal cells. We allowed them to grow and then we detached them. So, now what do we have out here? So, the surface now has the extracellular matrix of the young cells and the old cells. And now we put cancer cells on top. And we saw that cancer cells attached far better on the aged matrix than on the younger matrix. Okay. So, in other words, an aged normal and normal aged cell tends to secrete an extracellular matrix which is distinct in its composition perhaps, but whatever, but in its effect on the cancer cells, allowing for the cancer cells to attach, but not just attach, also move, because we did time lapse tracking of the cells to show that not just were the cells sitting there getting adhered better, they were not adhered at the same place, they would adhere and start moving around as well. So, there was a matrix that was more hospitable to adhesion and movement. Remember, I talked about two matrices again, laminin and fibronectin. I said that laminin allows cells to come together and fibronectin allows cells to move. We saw, okay, this is of course the metrics for the migrations and so on. We saw that there was higher level of fibronectin and higher level of laminin, both of which were being secreted by the senescent cells, that allowed cancer cells to attach better and move. And if one were to now just lay mixtures of fibronectin and laminin and put cancer cells, they would tend to attach better and start spreading better. Okay. So, and if one were to now use an inhibitory agent that would kill this adhesion of the receptors to that extracellular matrices, the cancer cell, the senescent cells, sorry, the cancer cell attachment to control extracellular matrix and senescent extracellular matrix comes and becomes almost equal. So, that tells you, yes, Ronnie. Yes.
So now does this sort of suggest a like two-pronged therapeutic approach where you give, so you have to reduce senescence while therapy is ongoing. So you have like you supplement some sort of extracellular matrix proteins into the whatever chemotherapeutic drug that is being provided. Will that sort of have a better prognosis?
Yeah. So this makes this finding very unpopular among the clinicians, of course, who tend to go with the let's just blast chemotherapeutic agents into cancer patients approach. This tells us that inappropriate and excessive use of chemotherapeutics actually makes things far worse in terms of
because you are enabling metastasis.
You are almost enabling metastasis here through excessive use of chemotherapeutics. But yes, but the sort of where the interventional aspect would be where we could perhaps use some of these, this is like what is called an RGD peptide. It's a peptide that prevents integrins from attaching to these matrices. So if you were to use perhaps some of these agents, you would prevent an excessive attachment of the cancer cells onto the matrix. One can also use, for example, one can use, what our work seems to suggest is it's important to also think about using what are called senolytic agents, agents that retard the senescence of cells in combination with chemotherapy to not just attack cancer cells, but also prevent some of these senescence inducing agents of chemotherapies by having some of these anti
Just out of curiosity,
senolytic.
what are some senolytic agents?
Resveratrol, the wonder stuff of vines.
This there was some other like weight.
Yeah, there are a bunch of them. There's one called berberin. There's one called taurine. I mean, there's an entire industry of age retarding agents that are there.
So these upregulate like cell autophagy.
I don't know what they do.
Maintenance stuff. Cell
I seriously don't know what they do. They are not they're not medicines in that sense. I think they come under the classification of nutraceuticals or supplements and so on. But yes, it's important. In other words, at least what we are saying is we have to take it seriously. We have to take the intersection of aging and cancer seriously. And if there are ways by which we can slow down the senescence of our normal cells, we can actually make them better defense barriers against cancers.
I think here, I mean, just as a very side note, the medical college coming up is a great avenue to test this as a
Yeah.
potential.
Sure. Okay. I have about 5 minutes left. Maybe I'll stop here and I'll take questions if there are, because there's an entire story and I don't think I'll be able to do that in 5 minutes. So wait, I'll just get to the. So this is on diabetes, which is interesting, but. Okay, so concluding sort of take-home messages. Okay, this of course is not relevant since I didn't talk about that. Okay, this is the one that I wanted to talk about. Oh, I didn't talk about this. Colonization. Aging stroma secretes an extracellular matrix that increases adhesion and migration of incoming cancer cells, and that could be one of the cell biological reasons why older tissues tend to do poorly when confronted with an onslaught of metastasizing cancer cells. Cancer cells spheroidal extracellular matrix, cancer extracellular matrix tends to enhance its elasticity. Incidentally, that deformation experiments were also confirmed with atomic force microscopy measurements, where we looked at both viscous modulus as well as elastic modulus, and we found that indeed these hollow spheroids were more elastic. The moruloids were more what is called viscoplastic in nature, and therefore it enhances durability in its travel across matrices. These are the two sort of take-home messages that I would give from two stories that came out of our group. Acknowledgements, of course, to students who did this work. The first story was almost led by my second graduate student, Jimpy, currently at Stanford, and then Tavishi, who's working for Intel, and Bharat and Mallar were the two students who worked on the second story with respect to how aging worsens the prognosis of cancer. Collaborators Deepak Proshenjit, who's a mechanical engineer in Sense, with whom we have created some of these microfluidic setups. Mohit, of course, has been a fellow conspirator for a very long time, and of course collaborators from our hospital, Annapurna. Tillman is a mathematician at Western Washington University who helped us with the cellular POTS model as part of the second story, and of course funders who funded some of this research. Questions?
When we say that chemotherapy might be increasing the basically metastasis, so we are just looking at the aspect of epithelial cells here. There are other aspects also that are basically affecting how the metastasis will be governed and the cancer cell proliferation will be governed. So in that sense, chemotherapy might be helpful in basically not because it is not inducing other aspects, then chemotherapy might be working there.
There's no question about that. You are absolutely right, and which is the reason why I said excessive use of chemotherapy. What happens many times is that when confronted with a case of cancer, clinicians tend to use one therapy after another of chemotherapeutic agents, where the usage is driven strongly by a very good level of intuition that the clinician builds up over time. In many cases, they don't have any choice. Radiotherapy cannot be given in the abdomen because we have so many different organs, and radiotherapy has its own problems. It tends to create fibrosis and fusions of organs. Suddenly you will start getting fusion in your abdominal normal organs will tend to suddenly close down. So, you will no longer have a hollow intestine. So, some of these because of these reasons radiotherapy cannot be given. So, you can only give chemotherapy. So, the problem what I was discussing is the fact that while chemotherapy is great in terms of its ability to kill the cancer cells, it can also have other unintended effects on the normal cells. And I also used the chemotherapy not as its not in its avatar as an agent of therapy, but also as an agent that can be used to make cells senescent. We have used other models of senescence as well, such as irradiation, which is often used to induce senescence, as well as used more aged cells by sort of cycling them more. So we have used several models of agent, and chemotherapy in that sense, or these agents are just one way of making cells older.
Do morula prefer binding to certain organs or certain regions of organs compared to others?
That's a very good question. So we've not looked at well, we know that in ovarian cancer spheroids tend to prefer certain organs more than others. For instance, they tend to metastasize to the liver to a far greater extent than say to pancreas and so on. The reason for that, we are trying to break it down into a bunch of more specific cues, like is the extracellular matrix more hospitable in the liver towards the metastasis, is the stiffness more hospitable towards attachment in the matrix and so on. So we have, there is a story there where we have looked at that certain ECMs are far better, far more hospitable, just like the senescence story. Certain ECMs are far better for moraloids to attach to them and spread, and certain, again, biophysical cues are very poor in terms of allowing the spheroids to attach. So yeah, we are looking at it from a more fundamental point of view.
So I had a doubt. So in case of chemotherapy, are the survival rates for morula and blastula the same?
No. We know that when patients come with the blastuloids, they tend to do much poorly.
So on the same lines of thoughts as the spore analogy, can we say that blastuloids are actually doing the same because they are much more resistant to chemotherapy?
Right. We have tested blastuloids and we know that they are more chemo resistant, and yes, that analogy can hold. But I mean, that analogy can be stripped down to its very basic extent and say that's something that tends to disperse and is resistant to any kind of attacks, tends to do better. So one doesn't need to necessarily pin it down to an ecological metaphor. It's a logical metaphor.
One more thing. So you talked about making the cancer cells unable to bind on the matrix, and I didn't get it. What happened? What was the relation then?
Sorry, in which context, if you tell me, I can explain.
So you talked about on senescent cells, say we make the cancer cells unable to bind to the matrix,
yes,
they become sort of equivalent to normal cells.
All that I meant was, if you now are able to add an agent that prevents the cancer cells from binding to those specific ECMs that the senescent cells make in excess, now the adhesion of cancer cells to both matrices becomes equal. Does that make sense? Yeah, that was a moot point.
So these senescent cells, they are able to make these two kinds of ECMs, right? I mean, one which is like more allowing the cells to kind of adhere, and the other it's like allowing the cells to migrate, right?
So what is these are just two of maybe several proteins that they are laying down.
Yeah. So when compared to normal cells, so what is different in the signaling that is happening that is allowing the senescent cells to make these kinds of extra kind of cell?
So it's not. We've not done that, so I don't know exactly what the signaling state of the senescent cells is. We have not explored it. We know, for instance, the cell biology of senescent cells is something Professor Deepak Saini, who's was a collaborator on this work with me, does to a far greater extent. So Deepak, for instance, has shown that senescent cells tend to have a very high levels of interleukins, for instance. So I mean, I think that there is a good amount of sequencing results in the literature, which can allow us to build a sort of a signaling map of senescent cells. However, those experiments have mostly been done with other cells, not with these mesothelial cells.
Yeah, I'm talking about like any pathways, like is anything different in the, I mean, the topology or networks?
There is a lot that is different, and it's known for other cells, not for these cells, and we have not done the signaling studies for these cells. So yeah, I don't know of what might be the altered signaling landscape.
And yeah, and then I mean, I don't know, like whether that could be like for a targeted therapy, like could we like target the senescent cells instead of like the cancer cells, like because they are, I mean, they are attaching to the senescent cells.
Agreed. And so that was the point I was trying to make with Ronnie as well, that one can therefore now think of using chemotherapy along with what are called senolytics. Now these are agents that specifically target senescent cells. So if you use agents which can kill senescent cells, now you have basically eliminated those attractors or attracting agents that attract the cancer cells to attach better onto these surfaces, right? So you're right. So that would be my sort of suggestion towards a translational ramification of these observations, that using senolytics along with therapy would go a long way in decreasing the kinetics of metastasis.
Okay, so if there are any further questions, I guess we can continue them over tea. So let's thank the speaker for a fantastic talk. So we'll break now, and so tea is outside.